Okay, so I think the session is open to the general participants. So I think I'd like to ask Singa to moderate the morning session.
Sure.
And if it is okay, let me start giving the opening remark about the general idea of this conference to the audience. So my name is Yongkun Park, the professor in physics, KAIS, and one of the co-chairs of the workshop with Seung-Ah Lee from Yonsei University and Lenji Zhou from Chinese University of Hong Kong. Let me first begin by express the gratitude to gracious host and all distinguished invited speakers gathered here. This is very multidisciplinary and the very collaborative topic on quantitative phage imaging. Last year, we had one conference on online format, and that turned out very successful, and we decided to continue to have the annual conference. But due to COVID situation, we are going to have the online conference this year. And today, we have 10 invited speakers from various the field. And we have two sessions. First, morning session, we have five invited speakers, and in the later part, we have 4 invited speakers. And recorded presentation for the speakers who has consented to distribute it will be posted on YouTube afterwards, so you can take a look at it. And one quick announcement before we start is that, as I said, we are going to have this quantitative phage imaging conference annually, and next year, hopefully, during the last week of August, we are going to have on-site conference in Jeju, 2 or 3 days, very tight, the face-to-face meeting. And we can also provide some opportunities to closely interact with the researchers between the students and professors. And we are going to have this conference in Jeju Island, one of the beautiful places in Korea. And all the conditions will be subject to COVID-19 situation. So we will keep you updated on that. And I hope that we can have the face-to-face meeting next year. Okay, so with that, I'd like to give it to the singer to moderate the morning session.
Okay, thank you, Paul, for the nice opening. Again, thanks everyone for joining us so close to the holiday season. Let's get started with the first speaker of the day. We have Professor Yibin Kang as the first speaker. Professor Yibin Kang is a Warner Lambert Park Davis Professor of Molecular Biology at Princeton University. And he's going to give us a talk titled Dact1 Biomolecular Condensates Mediate TGF-β WNT Crosstalk in Bone Metastasis.
Thank you, Thinga, and thank you, Paul, for the invitation to join this very exciting meeting. Even though it's virtual, it's great to gather everyone together. So I'm a cancer biologist, and the main focus of my lab is on the question of cancer metastasis. So for the last few years, we stumbled upon the curious phenomenon of phase separation, and I think our study is one of the first to demonstrate the role of phase separation in pathogenesis of cancer metastasis. So this is what I will try to share with you in my talk today. And in the beginning, I will just give a little bit of background because not many of you are probably familiar with the pathology or the molecular biology of bone metastasis. So as we all know, cancer patients usually do not die from their primary tumor. For example, breast cancer patients, if they have their breast tumor removed surgically and have no recurrence, usually they are cured. But if they have recurrence, oftentimes it not only could happen in the primary site in the breast, but often recurrence can happen as metastasis. And bone metastasis often is the first site of occurrence for a metastatic relapse. So about 70% of breast cancer patients have bone metastasis when they die from disease. And this is a really a painful complication for the patients because they could have fracture without having any fall or any accident. They could just have a fracture because the tumor grow in the bone, and that leads to a condition called osteolysis, and that will weaken the bone, and one day the patient might have a fracture for no reason. Or they could have severe bone pain or complications such as compression of the nerve, so-called, from the bone metastasis, and also the so-called condition of so-called hypocalcemia, because of the lysis of a bone release a large number of calcium molecule into circulation, and that could be life-threatening sometimes. So, of course, patients also die from other metastases like brain metastasis or liver metastasis or lung metastasis. But the main focus today is on bone metastasis. So if you look closer at a bone metastasis, this is actually an image, histology image of a human breast cancer cell line growing as bone metastasis in the mice as host of the disease. And you can clearly see that the tumor in the right side, they are growing, expanding, and they invaded into the space of the bone tissues in the left side. And so this is a complicated disease because it's not just the tumor growing. There are a lot of so-called stroma cells are involved, and they actually actively participate in the pathogenic process. So for example, the bone is actually not broken by the tumor directly. If you look closely, those cells lining the bone surface, they are not the tumor. They are stained in a darker red color, and they also contain multiple nuclei. These are the classical osteoclasts that we all have in our normal body. And we have them because our bone needs to be renewed constantly. The osteoclasts resolve the old bone and then allow the other cell type called osteocytes or osteoblasts to produce the new bone tissue. And so this renewal process happens every day, especially during the growth spurt of the adolescence. And that's important for the growth, but also for the maintenance of the strength and integrity of the bone tissue. What happens is when tumor invade into this space, they produce factors that stimulate the recruitment or the maturation of osteoclasts. And that leads to hyperactive osteoclast activity that basically alter the balance of osteoclast bone resorption and osteoblast-mediated bone building, and leading to osteoporosis condition. So there are also other cell types, as I mentioned. There are osteocytes, but there are also immune cells, blood vessel cells, and many other stromal cells involved in bone metastasis. So a lot of effort is focusing on trying to figure out a lot of crosstalk or the molecular communications between the tumor and the stroma as a way to either prevent or slow down the progression of bone metastasis, or at least minimize the painful complication of bone metastasis, such as bone, the weakening of the bone or resorption of the bone by the osteoclast. And that actually have led to a couple of drugs that are currently approved in the clinic to treat bone metastasis. One is called bisphosphonate, and the other one is an antibody against rankel called denosumab. Both of them have the same function of similar function of blocking the activity of osteoclasts, and that relieve the bone pain, bone fracture, and other complications, even though they actually do not really extend the lifespan of the patients. So there's still a lot need to be done to really slow down or revert the growth of bone metastasis. So one of the molecules that we have been very interested in the lab for a long time is a cytokine called TGF-β, transforming growth factor beta. So TGF-β is interesting because, as we know, when the osteoblasts build a bone matrix, not only they deposit a lot of the calcium phosphate and a lot of the organic and inorganic matters to make up the matrix, they also deposit a lot of growth factors, and they include a precursor of TGF-β that often become politically cleave and become active in the presence of protease produced by the tumor. So the bone matrix essentially store a lot of TGF-β. And there has been a lot of suspicion in the field that when the tumor invades the bone and activate osteoclasts to destroy the bone, TGF-β gets released into the microenvironment, and that might participate in this communication or the crosstalk between tumor and the bone stroma to promote bone metastasis. So one way to test that is to use an immunostaining called phospho-SMAD2 staining. So SMAD2 is a downstream signal transducer of the TGF-beta pathway, and it gets phosphorylated when TGF-beta binds to its receptor. And so phospho-SMAD2 in the nuclei is a marker of activated TGF-beta signaling. So what you can see is the tumor cells that are close to the bone surface have very intense phospho-SMAD2 staining, and that actually fits, or is consistent with the hypothesis that TGF-beta gets released from the bone matrix, and that signal to tumor cell to activate TGF-beta signaling, and that activate phosphorylation and nuclear organization of SMAD2. So that's a static image. We also perform a more dynamic imaging of the signaling event in live tissue of the mice. And how do we do that? So we engineer a cell line that encode two constituted stably integrated reporter. One is a radioluciferase under a constitutively active CME promoter. And so this is a constant and stable signal that will emit light when you provide the right substrate for radioluciferase. So that's an internal control. The other reporter is the firefly luciferase under the control of multiple SMAD binding sites, and that only gets activated when there are active TGF-beta signaling going on. So this becomes a dynamic reporter that will tell the strength of TGF-beta signaling or the abundance of TGF-beta in the environment. So we engineer the tumor and then inject into the mice, and the mice will develop bone metastasis after some time. So the first image we got is radioluciferase in the left side, as you can see. And you can see this mouse have two bone metastases in the left leg and in the right leg. And the strength of radioluciferase signal indicate the number of tumor or the size of the bone metastasis growing. And so you can see in the left side, this is the bigger bone metastasis, more tumor cells, and so stronger signal of radioluciferase. In the right side, it's bone metastasis that emerged later and the small one. But when you look closer by X-ray imaging, look at the condition of the bone, you can see the left side, the bone is mostly intact. But in the right side, if you look closer in the bottom, you can see there are areas of darkness. That's a sign of bone degradation, bone lysis. So even though it's a smaller bone metastasis, is an osteolytic lesion, very active bone lysis going on. So the expectation is this one should release a lot of TGF-beta and activate TGF-beta signal in the tumor. And that is indeed the case. When you do firefly luciferase imaging that reflect TGF-beta activity, you can see the right side, even though it's a smaller tumor, it have much stronger, more than a couple dozen times stronger relative TGF-beta activity, normalized by the radioluciferase activity, indicating that this is a much stronger TGF-beta response in the tumor because of the lysis of the bone tissue. And that also coincides, if you do a timeline imaging, you can see whenever you see this huge increase of TGF-beta relative TGF-beta activity, it always coincides with the detection of bone imaging. So this study, this is published many years ago, is a strong indication that indeed the hypothesis is true. TGF-beta gets released bone lysis and signal to tumor to activate TGF-beta signaling. And in the same study, we did a lot of the genetic and pharmacological studies showing that if you block TGF-beta signaling, you can slow down the progression of bone metastasis. And so we are interested about how does TGF-beta signal from the bone matrix to tumor promote bone metastasis. And this again is published, but the take-home message is that when TGF-beta gets released, it activates TGF-beta signaling in the tumor, and that leads to increased expression of a gene or a protein called JAG1, which is a ligand of a developmentally important pathway called Notch pathway. So JAG1 in the tumor combined to the Notch receptor in the osteoblasts or the osteoclasts and activate Notch signaling in those cells. And that have consequence because in the osteoblasts, that leads to increase of cytokines like IL-6 and CTGF, and that feedback to tumor to promote the growth of the tumor. in the osteoclast, pre-osteoclast, this signal directly increased the expression of a transcription factor called NF-ATC1. This is a master regulator of osteoclast maturation, and that leads to increased osteoclast activity, and that leads to more bone degradation and more TGF-β release. And so you can see this eventually constitute a positive feedback cycle. And so once it starts, it sort of fuels itself, and that drives the progression of bone lysis of bone metastasis. So it's clear that with the progression of bone metastasis, you should expect to see steadily increased TGF-β activity that drive the progression of bone metastasis. And this has been confirmed by many studies in the field. So because of that, we developed a antibody that specifically blocks this signal event between JAK1 and NOS receptor together with Amgen, and this has currently been tested to see if it could be used to treat bone metastasis. So that's about TGF-β signaling. The other one that we also is very interested is Wnt signaling. So just like TGF-β, Wnt is also highly conserved and developmentally very important for axis, formation or the polarity of the tissue or the body during development. And it's also called a functional use because Wnt signaling is really important for stem cell or cancer stem cell activity. So the role of Wnt signaling is actually quite complicated and could be controversial in the context of bone metastasis. So how we get interested in Wnt signaling is actually from the study of a cell surface receptor or the adhesion molecule called E-selectin. So E-selectin is a selective family protein that is expressed specifically in endothelial cells, and has been shown to be a very important factor to form the so-called perivascular niche for hematopoietic stem cells to promote the stem cell activity. And so that actually make it a very interesting candidate to study and test the hypothesis that when tumor disseminate into the bone, they might take advantage of pre-existing stem cell niche, such as hematopoietic stem cell niche, perivascular niche, to seed and then initially grow as bone metastasis. So we studied that to see if E-selectin is important for bone metastasis. This molecule actually has been suspected since its initial discovery in the 90s, I think. And so because it's one of the main functions is for leukocyte trafficking. So when you have a cut in a finger or there's an inflammation in the tissue, the leukocytes will be trafficking through chemokine attraction to the site of inflammation. And then they will use cell surface glycosylated protein that are ligands for the E-selectin to bind to E-selectin expressed in vasculature. And that leads to cell adhesion, the rolling of the cell, and then extravasation to basically get the leukocyte to fight infection. So if you think about it, that is very similar to how metastasis occur. You have adhesion, you have rolling and extravasation and development of bone metastasis or other metastasis. So we suspect that maybe E-selectin, or people in the field suspect E-selectin could be important for cancer metastasis. And so when E-selectin is cloned, people make knockout mice and then test to see if it can prevent the mice from getting metastasis when they have primary tumor. And so they test that, and the result was actually surprisingly disappointing. There is no phenotype or effect when people knock out E-selectin mice. The lung metastasis still develop normally in the high efficiency. The problem is those models only develop lung metastasis, and the mice never develop bone metastasis. So, so it's kind of forgotten for a long time until we thought that if you look closer, there's an interesting phenomenon that only in the bone vasculature that you actually see very high level of E-selectin expression, but not the vasculature in the lung or in the liver that often also have metastasis. So maybe people have looked at the wrong organ, the lung, because E-selectin is actually not highly expressed there. And so if you knock out in the mice, you're not going to see phenotype in lung metastasis. So we thought we should look at bone metastasis because E-selectin is specifically highly expressed in the bone vasculature. And so we did that. We crossed the E-selectin knockout mice with the not-skip mice so that we can transplant the human tumor to develop bone metastasis. And actually, when you knock out E-selectin, the mice indeed develop bone metastasis. By nuancing imaging, you can see less, by X-ray or micro CT imaging, and less lesion number when we knock out E-selectin, and less lytic area as well. So the genetic study shows that indeed E-selectin is specifically important for bone metastasis. The question is how does it promote bone metastasis? Bone metastasis is probably less dependent on cell adhesion and slow down the cell movement to promote adhesion and neutralization, because the blood flow in the sinusoid vasculature in the bone is very, very slow. It stands still. And also the vasculature in the bone is kind of perforated to facilitate trafficking of leukocytes during hematopoiesis. And so the cell adhesion function of E-selectin is probably not the reason why it's important for bone metastasis. There are probably some molecular cell biology change in the tumor when they engage E-selectin in the vasculature. So what we did is we performed a very simple experiment. We just cultured this tumor cell in a regular, cell culture dish, plastic dish, or in an E-selectin coated plate. And you can see that E-selectin coat, compared to the 2D monolayer of the tumor form is 3D sphere when they are E-selectin coating the plate. And they also re-express many of the typical cell surface adhesion markers that are typical of the epithelial cell, because this cell line that we studied is very mesenchymal, basal breast cancer. And so this is suggesting a phenomenon called mesenchymal to epithelial transition, the reversion of this called EMT, epithelial to mesenchymal transition. And MET has been shown to be important for metastatic colonization, similar to EMT have been reported to be important for initial migration and initial dissemination of tumor. The reverse process, MET, mesenchymal to epithelial transition, has in recent years shown to be important for metastatic colonization. So we look more closely in a global level. Indeed, we found that when the cell is cultured in the E-selectin coated plate versus the control, there is a positive enrichment of EMT down gene, the genes that are suppressed due to EMT, and there's a negative enrichment of the hallmark EMT gene. So this is basically suggesting that indeed there's a mesenchymal to epithelial transition happening when the tumor cell engages E-selectin. And more importantly, what we found is the most suppressed gene during MET is actually all encode secreted inhibitor of Wnt signaling, including DKK1, CTGF, and CYR61. So this is suggesting that the tumor is actually, when they undergo MET, when they engage the vascular E-selectin, they also downregulate Wnt inhibitor that potentially leads to increase of Wnt signaling to promote cancer stem cell activity. And that is indeed the case when we use a cell line that have a Wnt reporter, that have the TCF binding site. You can see that without E-selectin coating on the plate, only when you treat the cell with Wnt3A, you have activated reporter activity, indicating increased Wnt activity. But if you culture the cell in the E-selectin coated plate, even without Wnt addition, you already have Wnt signal activation, and that is further increased by Wnt3A addition. In vivo is also the case in the area that have high density of vasculature, as you can see in the second panel. Those are the area that have the strongest Wnt reporter activity in vivo in those bone metastases. So this suggests the following model that we know tumor needs to undergo EMT to migrate and disseminate. There are many signals like TGF-β could induce EMT. We actually don't know how MET is induced when the tumor disseminates to distant organ. Now we have a mechanism. When the tumor binds to E-selectin, they activate MET, and at the same time, they allow Wnt to be hyperactivated by downregulating Wnt inhibitor. So this allows the cell undergo EMT and at the same time maintain cancer stemness by increased Wnt signal. This is important because Bob Weinberg have shown that EMT promote cancer stemness. So supposedly when the cell undergo MET, they should lose stemness. But by engaging E-selectin, they can undergo MET and at the same time still maintain high cancer stem cell activity, which is important for bone colonization. So the study shows that at least in the initial seeding in the vasculature, Wnt signaling is activated by E-selectin engagement, and that is promoting bone metastasis formation. But there are also studies in the field, actually one is done by my postdoc mentor, John Massaguet, and the other one is done by my former postdoc, Guohong Hu in China. And both studies in two different angles shows that Wnt signaling actually has to be suppressed to allow full-blown bone metastasis to develop. In one aspect is if the cell have active Wnt signaling, they are proliferated and they attract the attack by the NK cell. So by producing high level of DKK1, which is a Wnt inhibitor, those disseminated tumor cell will escape from the immune killing by NK cell. And this is important for the expansion or the long-term dormancy of those bone metastases. In the later phase of bone metastasis development, tumor cell also needs high level of DKK1 because this is used to suppress Wnt signaling, because Wnt signaling promote the osteoblast to express a cytokine called OPG, which suppress the rank L induced osteoclastogenesis. So by suppressing this loop, the tumor cell can activate osteoclast activity to drive bone metastasis formation. So the bottom line is that in both cases, the Wnt signaling in the later phase of bone metastasis development need to be suppressed. So to summarize what I've talked to you so far, during the bone metastasis development, we believe that there is an increasing TGF-β activity because TGF-β gets released from the bone matrix, and that is driving bone metastasis development. But there's a switch of Wnt from relatively high in the beginning, in the initial seeding to the vasculature, to later on in the later phase, there need to be a decrease of Wnt signaling. And so you can see the cell is switching from low TGF-β high Wnt to high TGF-β low Wnt. And this could be happening completely separately, or it could be somehow coupled with some kind of central regulator that couple these two signaling pathways together. And so that leads to the main focus of my presentation today, which is the importance of a molecule that is involved in the coupling of these two pathways. So the insight is from a lot of work to derive a bone metastatic subline from a breast cancer cell line called SUN159. And to make this long story short, what we did is we isolate a so-called M1A cell line, which can develop bone metastasis with high efficiency compared to a primary parental cell line. So you can see that after TLV injection, only the M1A develop bone metastasis. So if you compare the highly bone metastatic variant compared to the parental or the weakly metastatic line, you can see similar to the system I told you before, is a strong enrichment of TGF-β activity. So this is consistent with what I just told you. And so we also look closely at the genes that are highly expressed, specifically in the M1A line. And we are particularly interested in Dact1 for a couple of reasons. One is that this gene has been reported to be a downstream target of TGF-β signaling. So this could be induced by TGF-β signaling. And also there are some conflicting reports that showing Dact1 could be regulating Wnt signaling. And so this could be a regulator that is sort of coupling these two pathways together for that kind of switch that I mentioned earlier. So we confirm in our own hands that indeed, if we treat a cell with increasing level of TGF-β or with long exposure of TGF-β, there is increasing level of Dact1. So this is indeed induced by TGF-β signaling. And in a cell line that is deficient in TGF-β pathway, there is no such induction. And also interestingly, further increasing our interest, we found that when the patient's tumor have high level of Dact1, they have high risk of developing bone metastasis, and specifically only bone metastasis, but not metastasis to other organ sites. So this indicates that in clinical situation, Dact1 could be a downstream gene of the TGF-β that is driving bone metastasis development. So the next question is, how does it really involve in the regulation of Wnt signaling? So what you use is, again, using a Wnt reporter. And if you treat the cell with the Wnt3A, you have increased Wnt-reported activity as expected. And then when you use the inhibitor, it will block that. So what we did is compared to control in the middle, either we have Dact1 knockdown, you have a stronger Wnt response, or Dact1 overexpression, you have a weaker response. So this suggests that Dact1 is, in this system, in the tumor cell line, is the inhibitor of Wnt signaling. And so indirectly, we can also use TGF-β to control Dact1 activity and see how that affects Wnt signaling activity. So when you treat a cell with TGF-β that increase Dact1 expression, you have less Wnt response. When you treat a cell with Wnt38, less response. When you use a TGF-β inhibitor, you have reduced Dact1 and then increase Wnt response. So what this tells us is that TGF-β induce Dact1 and Dact1 suppress Wnt signaling. And so therefore Dact1 could be a intermediate that mediate the switch of height increasing TGF-β and then decreasing Wnt. So the question then is, how does Dact1 regulate Wnt signaling? And so usually you will look at the localization to see where is Dact1 located. And to our surprise, when we fuse Dact1 with TD tomato, either in the C terminal or in the non-terminal, the fusion protein actually expressed not as a diffused protein in cytoplasm. It's actually forming a very interesting, curious punctate structure, or possibly condensates in the cell in the cytoplasm. And this is not just an artifact for a fusion protein. If you use a native antibody against the native Dact1, you can see the native Dact1 also forms those condensates. And if you knock down Dact1, those condensates disappear. And those punctate structure also could be detected in vivo in the bone metastasis. You can see the Dact1 TD tomato in the tumor cell could be detected in forming those condensates in bone metastasis. So here then we use the TomoQ technology that you'll hear a lot today to perform hollow tomography analysis to see how are those TD tomato Dact1 condensates localized and what's the biophysical property. So you can see in the area of increased refractive index, those areas co-localized with TD tomato positive condensates. And the refractive index is about in the range of 1.36 to 1.38. So that is consistent with the typical refractive index of a phase separation, biomolecular condensate. We also use fluorescent recovery after photobleaching to show that there's a partial recovery of the bleach fluorescence, suggesting that is a dynamic organelle that have both mobile and immobile components. So is this a new condensate or previously reported one? For example, it's been reported that Axin-1, which control the stability of the Wnt signaling complex, can form a condensate, form condensates in the cell, in the cytoplasm. And so when we do immunostaining, we show that the Dact1 TG tomato condensate in red is completely different from the Axin GFP condensate. So these are completely different type of condensates that both could be regulating Wnt signaling activity. We also look more extensively at other either membrane bound or membranous condensates, organelles in the cell, including Golgi, ER, or phagosome, lysosome, endosome, P-body, and mitochondria. And invariably, we show that the TD tomato Dact1 positive condensate is completely different from the other previously reported organelles. So this is a completely new one that has never been reported before. So as you know, the biomolecular condensates are usually formed by this so-called intrinsically disorganized, proteins with multiple intrinsically disorganized domains. So what we did is we used D2P2 software to predict that the Dact1 protein indeed contain potentially up to 10 distinct disorganized domains, including a relatively large one in DR7 and DR8. So what we did is we delete those domains one by one and see if the deleted version of the protein can still form the condensates. So the result is the following. So wild-type protein form condensate, and a lot of deletion means still form condensate, but DR7 and DR8, the two that have very large disorganized domains, when you delete them, indeed the cell, the fusion protein can no longer form the condensate. So then this will allow us to test whether the condensate formation is really important for Wnt pathway, Wnt signaling suppression. So what we did in here is we transfect the cell with either wild-type Dact1 protein or the DR7 deletion mutant that no longer can form the condensates. And then we treat the cell with the Wnt3A to induce the Wnt reporter, TGF-B reporter activity. So you can see that in the cell that have transfected with Dact1, but some of them are not transfected. Is not 100% transfection. So some of the cells have very few transfection and they don't form the condensate. Those still have Wnt reporter. But whenever you see the weak or losing of Wnt reporter activity, those cells have condensate formation. So this shows that wild-type protein indeed suppress Wnt signaling, and only when those cells that do not have condensate will have active Wnt signaling going on. But the mutant that cannot form the condensate can no longer suppress Wnt signaling. So the question then is, how does this condensate regulate Wnt signaling? And so what we did is we decided to look at the protein composition of the condensate. So we developed a method to use mild detergent plus sonication to essentially break up the cell, but keep the condensate intact. And we can actually use flow cytometry to isolate the TD tomato positive particles from those lysates, and then perform mass spec analysis. So you can see in the Wnt treated versus Wnt untreated condition, there are a lot of proteins that could be isolated from those condensates, and some of them change the concentration. But some of the most abundant proteins, of course, we find the Dact1 in the condensate, but we also find all the subunits of the CK2 kinase, which is a very important kinase that support or activate Wnt signaling activity. So the hypothesis is that the condensate could sequester CK2 from cytoplasm, and that prevents CK2 from supporting the activation of Wnt signaling. So we look at that. If you just transfect CK2 into a cell, it's mostly localized in the nucleus, but also there are a lot in the cytoplasm, and this is the compartment that is important to regulate Wnt signaling activity. But when you co-transfect CK2 GFP with Dact1 TD tomato, the Dact1 form the condensates, and you can see that in the cytoplasm, a lot of the CK2 is then concentrated, sequestered in those concentrates. So we can also see that by flow cytometry. So here, if you just simply transfect TD tomato with CK2 GFP, you can see that there's no tomato enriched in the condensate because they don't have those disorganized domain as expected. Now, if you co-transfect Dact1 TD tomato with just GFP, the TD tomato Dact1 will be localized in the concentrates, but there's no GFP because GFP does not bind to Dact1 and localize in those concentrates. Then when we co-transfect Dact1 TD tomato with CK2 GFP, now you can see that TD tomato, as expected, localized in the condensates. And you can see that the large majority of the GFP is now also contained in those condensates together with TD tomato Dact1. So this suggests that when the condensate is formed by Dact1, it also sequesters the CK2 GFP. So the next experiment is that whether the ability of Dact1 to suppress Wnt is mediated by CK2. So how do we do the experiment? So you can see that when you treat the cell with Wnt3A, it will activate Wnt signaling. But then when you use a CK2 inhibitor, it will suppress Wnt signaling activity. But in a cell that have high level of Dact1 expression, overexpressed Dact1, the baseline level of Wnt signaling response is decreased. And also furthermore, when you try to inhibit CK2, there's much less inhibition of signaling because CK2 is no longer involved in supporting Wnt signaling because it's taken away by Dact1. So this experiment supports that the main function of Dact1 is in suppressing Wnt is going through sequestration of Dact1, CK2. So finally, we did an experiment to see all this thing with all this information. Can we reduce the development of bone metastasis by targeting Dact1 and eliminate it to disallow the switch of TGF-β on Wnt signaling in the development of bone metastasis? So create two cell lines with knockdown of Dact1 compared to the parental control cell line, and then inject in mice. You can see that compared to the parental line, there's a nearly tenfold decrease in one log scale decrease of bone metastasis development by imaging. You can see that in the mice as well, and there's less bone lesions and less lytic area. So this is all supporting the role that the following hypothesis that during the progression of bone metastasis, the increasing level of TGF-β induce the Dact1 expression, and that leads to increased CK2, and then Dact1 expression, and then that leads to increased sequestration of CK2 in those condensates, and that suppress Wnt signaling. And so this allows the transition of the tumor cell from a low TGF-β high Wnt to high TGF-β low Wnt activity. So with that, I want to thank the student who did the work, especially Mark Esposito, who did the work on the Dact1, and he's the one who take advantage of the TomoQ demonstration in our department and use the equipment to obtain a very critical image of the high increased refractive index of those condensates. And that's a critical experiment to lead us to identify this new phase-phase separation phenomena mediated by Dact1. So with that, I'll be happy to take your question.